So thank you, Martin, very much. And it's my great pleasure to be able to talk to all of you and reach to the attendees of the DMPH 2019 conference, albeit remotely. But as one of the co-organizers, I would first like to thank Martin for his leadership role in this organization, along with, of course, the other co-organizers, Dr. Chandra and my good friend Carmen. And of course the ICTS organization and their extremely capable organization and funding for kind funding for this conference. And a very good morning to all. It's of course night here, close to midnight actually. So I reserve the right to yawn when I have to, but I'll just get started. I will be describing some methods for mathematically characterizing in the form of a hierarchy the high-dimensional human immunophenotypic diversity that we see in many studies which are related to biosecurity. I'll go to a few examples, but the most of the talk will try to cover the models and the motivations behind this research. Okay, one of the easiest ways to understand where this research comes from is the lab research directions, which has been kind of been there for past more than, I would say, 15 years now. And the lab works in two orthogonal directions, but sometimes they're overlapping. So along the horizontal direction, you see that we work with high throughput omics data and we work with high resolution models. So these are platforms that we were among the earliest teams to develop at initially at MIT and then Harvard Medical School and then in India and then back in Pittsburgh. So these are mostly very high throughput data sets, so they can allow you to observe phenomena at fairly great detail. So they're high dimensional by definition, but they can also give you high resolution measurements, especially at the level of single cells. And we were again one of the early teams who started working in this in more than a decade ago. And so some of the data sets and applications for today's talk come from such single cell level data. In the vertical axis, you see our interests in mostly biosecurity and big data problems. We are interested in working problems where we characterize the immunophenotypic diversity, mostly in humans. We look for biomarkers and then we try to make predictions. We are also interested in big data in public health. You must have heard already from some of the methods that are used in our lab from my colleague, David Sinclair, who has presented a couple of days ago. And so we are interested in such population models, either using simulation, data fusion, data integration, and so on. So the common theme which ties these two approaches are, I would say, modeling of hierarchies of individuals. So this is the common theme in almost all my projects. So we look at data at fine resolution, where the individuals is a generic term. They can be defined in the terms of individual molecules and markers to individual cells to going up to individual humans or even individual subpopulations and cohorts. So as soon as we model a hierarchy of these individuals, and I'll show you an example of that in today's talk, of course, these individuals could be observed across different levels of this hierarchy. So then we are in a position to study biosecurity issues, and different kinds of biosecurity questions can be addressed. So why do we study diversity of human immuno-phenome for understanding biosecurity at a systems level? That's the motivating question. So the original motivation of our studies came from when we looked at the more and more of the land use data and urbanization in the 21st century. We started looking at this from a spatiotemporal modeling point of view, and these projects are ongoing, especially with the Indian land use data. My lab has been working on this for quite some time. So, of course, global urbanization, when you look at it as a percentage of national population, India and South Asia in particular, doesn't look like more than 30 to 40% urbanized. Yet, if you look at the projections of the UN, and if you look at the projected growth in urban population in developing countries in the coming decades between, let's say, 2010 and 2050, the largest projections come from India and China. Not surprisingly, almost 500 million people are expected to live in cities by 2050. Now, why is that a biosecurity concern? It's because of the concept of so-called unhealthy landscapes. So this was a term which was initiated in a paper called Unhealthy Landscapes, How Land Use Change Affects Health. So this was a result of a convention in 2002 in Washington, D.C. by the International Society for Ecosystem Health. It was noted that land use changes are the primary drivers of a range of infectious disease outbreaks and emergence events, and also modifiers of the transmission of endemic infections. Often such land use changes could be rapid, leading to urbanization, deforestation, biodiversity loss, wild habitat encroachment, farming, mining, logging, etc. And of course, that leads to exposures of hitherto unexposed environments. So therefore, now we have the so-called One Health Challenge, whereby you have emergent human-animal environment interfaces. These are new interfaces that open up and can present new biosecurity challenges. We have seen this in one after another zoonotic disease outbreaks across the world, including the Nipah virus outbreaks that you have probably heard of in certain states in southern India this year and last year. Threat of emerging and re-emerging zoonotic diseases seem to grow. Unfortunately, they've been growing steadily since 1940s. The incidences are higher every year, even if you control for effects of higher reporting in the later decades. It has been found that 60% of the known human infectious diseases and almost 3/4 of newly emerging ones are of zoonotic origin. That is, they often originate in vertebrate animals first before being transmitted to humans. If you take a conservative estimate, the number of viruses present in such zoonotic transition could be 1 million, which means almost 99.9% of the viruses that lie out there are currently unknown to humans. So the threat is large. How is the threat distributed? If you want to look at the global distribution of relative risk of an emerging infectious disease event, whether it's due to zoonotic pathogens from wildlife, from non-wildlife, from drug-resistant pathogens, from vector-borne pathogens, then we see that India kind of blows up all the time. And so I guess the researchers have the onus to address this problem, and especially public health researchers. The model that we adopt is an integrative one. We try to get to sources of data on pathogen, the host, and the environment. There are a variety of different types of data sets which are available, sometimes not available, sometimes with missing data, sometimes with not adequate design. So with all those flaws, different types of data sets are available, especially for the more popular agents that cause disease. And we try our best to do data integrative modeling, mining, mapping, clustering, and prediction using so-called big data techniques. So that's how the lab works. So in order to address some of the key questions in biosecurity systematically, I'll give you the examples of such questions that we address mathematically. So first is, how do we mathematically characterize a normal subject's immunophenotypic profile? So this is really, really important, because nobody typically would like to pay you for collecting normal subjects' data. Yet, if you do not have a very good understanding of the diversity in the so-called normal population, then you would never be in a position to, with a high confidence, be able to detect what is a departure or a deviation from normal. So secondly, how to model a diverse range of such population immunophenotypes, because they would typically be distributed in a way in which you would have so-called small area estimation problems. You will never have adequate representation of any particular subgroup in a finite sample. Thirdly, how to group those population immunophenotypes into meaningful classes. So this is again a clustering and classification problem, because the immunophenotypes will not probably be having, will probably have meaningful classes which could be used for answering specific hypotheses. So unless you have classified them, you would probably not be able to and do the downstream analysis easily. And then there is a spatial problem, how to map those immunophenotypic classes over geographical space and time. Then how to assign correct classification to new or rare immunophenotypes so that they don't get overwhelmed or absorbed by the more common phenotypes. Then how to rapidly detect any departure or outlier from a given range of normal profiles, how to represent population profiles in databases to enable mining and fast queries. This is a computational efficiency question. If you have a lot of diversity of phenotypes, in order to match them, if they're high dimensional phenotypes and you do not have a data standard or a data format, this can become computationally difficult and time-consuming challenge. Then how to find associations between population genotypes and immunophenotypes. So this is a more classical question in genetics. So you are trying to find associations between genotypes and phenotypes, but in this case, immunophenotypes could be quite complex. They're just not one variable, but they could be high dimensional. How to identify a vulnerable or resistant subpopulation based on such associations. This is probably the most important question you have, where you want to probably intervene and predict an outbreak or do something about it. So you want to know who are the people, what's the cost attached to addressing the problem in that particular population. And then finally, we have the bigger question of how to systematically estimate the risks and parameters of potential outbreaks in a given population. So this comes more from the health economics perspective, but it's nonetheless for the health policymakers, this is the most important question. So all of these we know how to answer, sometimes more, sometimes less, but we have some ways of answering these questions. But today, of course, I will restrict myself just to show you some methods, new methods in particular, how we study the diversity of the human immunophenome. The data has three properties, and they are high dimensional data, which means a lot of markers are going to be observed at the same time. They are high throughput data, that means the markers are observed in a large number of cells. And they're high resolution data because we are going to make these data observed at single cell level. So in this particular study, the data comes from flow cytometry. It doesn't have to be just this platform. There are other platforms which are equally useful. But for the present data set, it's high dimensional, it's multi-parametric, which means a lot of markers are used to define or determine every individual subject's immunophenotype. The number of markers can easily be a few dozens. You are studying individual cells, and therefore the number of cells in any given sample drawn from an individual subject could be somewhere between tens to hundreds of thousands, sometimes millions. So the cells are in the rows of the matrix. Every event is a p-dimensional readout of p markers in parallel, so you can easily have 1 billion observations per sample per person. And if you have a large cohort, then you are looking at 1 trillion data points per study. So that's a big data problem. The problem is getting even more challenging because you can measure more and more markers these days. The earlier constraints based on the color spectra of fluorescent tags are getting improved by using instead stable isotopes of metal ions, which are read with mass spectrometry. And so these are so-called mass cytometry machines from which we collected the data for this present study. So what I am trying to answer is, how do I characterize a high-dimensional multi-parametric immuno-phenotype of a given subject? First, we will do it for one subject sample, then we will do it for a cohort of such subjects. So the way we approached it about 12 years ago, we started looking at this problem. We wanted to mimic the fact that a complex immunophenotype is complex because of the presence of populations and subpopulations of cells showing a variety of characteristics in high-dimensional marker space. So since it's a mixture of such subphenomena, we wanted to capture this using mixtures, newly defined mixtures of probability distributions. The usual mixtures were not good enough to capture the dynamic and the stochastic nature of these phenomena. Not that they were wholly inadequate, but we needed to look into more spatial and temporal phenomena. So that's why we needed to design new mixtures. The idea is simple. The immunophenotype for a given subject is the overall distribution of its points in the high-dimensional marker space, as you can see in this three-dimensional points. This is high-dimensional data. I can only show you 3. So here, each dimension corresponds to the expression of one immunologic marker. Each point in this plot represents the expressions of markers, P markers by a single cell. I'm just showing you 3 for this picture, but it's actually high-dimensional, P-dimensional data. Such expressions of immunologic markers could often be stochastic and noisy. In other words, each cell is kind of free to express its own markers, and the noise comes from the platform and the measurements therein. And therefore, there is significant diversity of immunophenotypes as a result of this dynamic, stochastic, and noisy behavior of data. So this presents a serious statistical modeling challenge. We often see multiple modes, so one distribution never suffices. We often see heavy-tailed phenomena. There are lots of outliers of points which are far away from the center of the distribution. And we often see tails, which are skewed multivariate data, which means they are tails, but they're not even tails along one dimension, but multiple dimensions at the same time. So these properties are, as annoying as they tend to persist even after standard data transformations are applied. You cannot wish them away. You cannot transform them away. So you have to take care of them in your model. So you can see the model is shown in the plot below. So that's the model of the cell populations that appear in the plot above. So you can see that the contours of the distributions or a mixture of distribution that's used. I can tell you that there are 5 cell populations, and the cell populations have a mean location. They have a certain shape, a certain variance, a whole lot of parameters, including parameters for skewness. You can see they have spatial properties, like some cell population is tilted, some cell population is shrunk. So all of these properties are resultant of the expression along these different dimensions or different immunologic markers. So in other words, if you change the condition, for example, if you did a phosphorylation of the cells, then along the SLP76, which is a phosphorylation marker, suddenly you will find these distributions now have different parameter estimates. They might have changed shape, they might have changed position, they might have changed size, they might have changed tilt, they might have become tight or diffuse. So all kinds of ways in which they could mathematically, they could change, which can be captured using the mathematical model. So we have created several platforms to suit different suites of platforms, programs that try to capture different mathematical transformations that take place in these immunophenotypes over our research in the last decade. So today I will mention a recent paper that was submitted from our team and that addresses a very specific computational challenge. I will give you the references to some of the earlier work, but today's description is for a very specific challenge, and that is how to do rapid detection of rare events in a complex immunophenome. So give you a good model to specify or describe a very good model is possible often when the data is comprised of cell populations which are relatively which in terms of their size are relatively comparable to each other. This is often not the case when you are interested in emergent phenomena, which typically emerges in the tail of many so-called non-clonal cell populations. So those are cell populations which start acting differently, and they start acting differently often at the tails, and it's almost in the flavor of speciation, as if a new species of a subpopulation emerges and then becomes its own cluster, its own group. So to capture the dynamic emergence of such rare events, it's very important. For example, they may be drug-resistant cancer cells, or they may be a very small subset of circulating tumor cells in blood. So they have a variety of applications in biomedical studies. So we want to capture them, and the difficulty in capturing them is because this is high-dimensional data. The data shapes are very complex, so those rare populations may be actually hidden under different shaped populations, and so they're not easy to detect. And the most important problem is that because of the so-called curse of dimensionality, so in a high-dimensional data, you have exponentially increasing volume, exponentially increasing in the number of dimensions. So you don't know where to look. If you know where to look, then you can zoom in. You can set do gating methods of zooming in, setting thresholds and zooming into places where you expect to find those cell populations. But if it's a dynamically emergent cell population, you don't know when and where it emerges. So you need a method to still do the search space, this extremely large search space, and find that rare cluster and be able to still validate that it is not a fluke, it is not a chance finding. So you have these two concerns. One is that you don't know where you're going to look. It is somewhere, but wherever it is, it keeps on occurring in that place in multiple samples or replicates. So there is some way of biologically validating that rare find. Okay. So to this end, we built a novel hierarchical Bayesian model to share information from multiple samples. So you need the samples to talk to each other, because when you find these rare cell populations, somebody has to do like a vote counting to make sure that across samples there's agreement that this is a rare population, but it is a real population. First, we model cell populations in each sample by a mixture of probability distributions. We have used different families. Let's say over here we will describe Gaussian components. So these are Gaussian clusters. But so we need, of course, as soon as you describe the mixture, you are going to get an assignment of a probability score to associate each cell to a given population. In this case, a Gaussian distribution. So that reflects the underlying differences across individual samples. So that's a bigger picture. But now you come to the second part, where we let the Gaussian components corresponding to cell populations in different samples be similar to a common prototype populations up to a certain small perturbations. So modulo some amount of movement in space, we allow the populations to match themselves against each other across samples, so that we can then capture the minor differences which are not accounted for by this movement between individual biological samples. So those are the kind of populations we are interested in. So third, we allow that some Gaussian components appear only in certain samples and output these populations even if they are rare. So these are the three steps in which the algorithm proceeds. The data likelihood is given in this case as a mixture of Gaussians. So let the cytometric data be denoted by X and the cell memberships by the matrix H, where H M sub nk denotes the membership of the nth cell in the mth sample to that particular sample's kth Gaussian component with mean mu sub k and precision lambda sub k. So the data likelihood is, of course, defined in terms of all the data points. In this case, the data points come from Gaussian densities, and each data point xn goes from n equal to 1 to n sub n, where n sub m is the number of cells in sample m. They come from k Gaussians, so those are the density distributions parameters. In this case, there are k densities. And remember, k is the number of Gaussians which is common across all the samples. So that comes from a prototype. And if any particular sample has more Gaussians or more distributions, then they will be that will be specified in my next slide as a very specific probability vector. M, of course, is the number of samples. So that's how we get our set of parameters given by theta, and each data point in this case has dimension d, which is therefore a d-dimensional Gaussian distribution. So now in the hierarchy, not only are the points distributed, the populations themselves are distributed in certain ways across the different samples. So that is given by that latent membership indicators H, which has a factorized discrete probability distribution, and it essentially is defined in terms of the probability pi m sub k of the kth population appearing in sample m. Of course, they sum up to 1, but some of them can be 0 because some population may or may not appear in some sample. So overall, the prototype makes sure that there is agreement of most populations across samples, but some population of cells may or may not exist in some samples. Okay, and then to model the uncertainty in specifying this probability vector, we assign a symmetric Dirichlet prior distribution over it. So the common prototype model is shared among the different samples. We allow cross-checking for the validity of the detected rare events across this common prototype. To share information between clusters of different samples, and we used parallel message passing algorithm to make this to do this fast. So this is actually sharing of information where each sample is being clustered in a different machine. So we let the mean parameter mu of each cluster in a sample follow a Gaussian prior distribution common to all samples, and then the distribution is conditional on the parameter eta, which is estimated as from the data just as mu, and beta naught is the other hyperparameter in the model, which allows you to get the variance or the precision. Similarly, the covariance matrix is given by lambda, which follows a Wishart prior distribution. So these are fairly standard techniques, and we follow those just to make them computationally efficient. The prototype components have to be also, we need to estimate the parameters of the prototype clusters from the data. So we assign a Gaussian hyperprior distribution over the mean of each prototype cluster. In that case, in the earlier slide, I was mentioning eta k, and we assign an inverse Wishart hyperprior distribution over the shape of each such prototype cluster, and then the model is complete. So this gives us a joint probability distribution over the set of parameters and the data set X. and this is given as a product of the parameters and the hyperparameters which are specified as per the given graphical model that's described in front of you. So a recent application of this model was based on the complex multi-parametric immuno-phenotypes that came from a 13-dimensional mass cytometry dataset. The dataset was generated by Levine et al. 2015. It used manual gating by a human expert as a gold standard way of characterizing the cell populations, the hierarchy of populations present therein. It contains measurement on 167,000 plus cells derived from a sample donated by a healthy human individual. How does the healthy human immunophenotype hierarchy appear? This is bone marrow data, by the way. These data are difficult to analyze due to the large variation in the cell population hierarchy. It was observed by different researchers that the smallest population is a platelet population consisting of just 5 cells, while the largest is a population of almost 14,000 mature CD4 positive T cells. So distinguishing such pebbles from boulders is computationally challenging, because when you train your model to go top down, then they tend to pick up boulders easily. When you train it bottom up, then they pick up the pebbles easily. But when you want your algorithm to have dual capacity, it's a challenging problem, especially if you view it as clustering, because if there are n points, there's order n square possible distances in this clustering, and you can see that there's fairly large data sets. So this is like this. So you can't really do a hierarchical clustering approach and be able to solve it by handling order n square distance matrix. So this was the result from our study. We found the 24 different cell population in the hierarchy. We used our model to solve the, also we predict a class label for each cell, thus solving a classification problem. We assessed the performance of our model and by comparing it to the predicted cluster labels, which were predicted by, which were available by manual gating, which is the gold standard in the field. Then we calculated the F-measure score to see how our classification performed. It's defined as the harmonic mean of precision and recall, which ranges between 0 and 1. So our model achieved the F-measure of 0.7 for this particular data set, outperforming previous results reported on this data. It was observed that all other considered methods performed somewhat poorly on this data, achieving low F-measures. This is a summary, a visual summary of the results. The first column tells us for the performance of our method, and the other columns are for other known methods. And our method gave a fairly good classification for almost for 3/4 to, I would say, 70% to 80% of the 24 cell population. So here each row is a cell population. Also noted that there are irregular and skew-shaped clusters. A large number of them have variations in their skewness, and we are happy to note that we were among the early teams who developed different new skew normal and skew T families, which gave rise to a lot of active research in the field in the past 20 or so years. So our skew normal and skew T mixtures were the first of their kind, and we have applied those subsequently. So this is a partial list of the work that we conducted in this area. The first paper is the work that I described, the model that I described is called FLARE, or Flow Cytometric Analysis of Rare Events. It's been submitted in bioRxiv now. If you want to give us feedback, feel free and feel welcome to do so. The work was done in collaboration with scientists from Purdue, from Yale, from Harvard, from Singapore, from Memorial Sloan Kettering, and us in University of Pittsburgh. And I will end with a summary of the modeling pipeline that we are using in many problems, and we tend to get nice results. If you're interested in collaborating in different aspects of this pipeline, feel free to contact me. So the pipeline has 5 components, 5 general components. In the first step, we collect or generate multivariate data streams on health events through diverse mechanisms like sensors, omics, EHR, unstructured imaging data, etc. In the second step, we construct an integrative model in high-dimensional space of which only some of the dimensions may be observed by each individual data stream. This is the place where we do data fusion or missing data problem. So when we have solutions to the data fusion problem, we can combine an optimal set of high-dimensional features that can jointly describe, using a so-called statistical joint distribution, of overall profile of health of a system. It could be immunologic health, physiologic health, metabolic health. If it's omic data, then omic version of health. Then we train and fit suitable multivariate probability distributions, as you just saw, to let the model parametrically characterize the baseline concepts of a system's health. So, for example, if I know the position or the skew or the shape of a particular cell population, of all cell populations, and I know this for a whole cohort of subjects, then if there is a new person showing outbreak of a disease which is not characterized in any of those parameter sets, then I can detect that very easily. So we detect drift in such concepts of health based on statistically significant deviations in the system's model parameters to detect individual outliers. A good example, conceptual example, came from a 2012 paper where we tried to outline this concept, where you can use surveillance of phenome information to detect outbreaks. So typically the phenome is high-dimensional data which is modeled, which appears like this. I'm not showing you high dimensions, but this is real data. You model it, and then you know the relative distribution of all the cell populations. You know where they tend to congregate, where they do not. So you have an idea of the different cell populations and their orientations. Then you can model this in high-dimensional space, and you can see, you can do even a dimensional reduction techniques where you can capture this information as a curve going from high to low density regions, going from peaks and troughs. So in other words, you can take the high dimensional landscape of a phenotype and reduce it to curves. The reason we do that is then we can match these curves for a large number of patients, and very quickly this mathematical descriptions allows us to A, search and query databases of such representations, and B, model outliers, and C, that if there are certain subjects with immuno-phenotype which diverge a lot from the mainstream. In other words, this gets reduced to a problem of clustering and classification of functional data. So thank you for your kind attention. If you are interested in any of this, or if you have your suggestions, or if you think you might be keen on collaborating, please feel free to contact me at that email address. Thank you.